welcome Helmi Kreinin to the stage.
Hi, so my name is Helmi and I'm a chemist. And I'd like to tell you a little bit about how we go from the information you inherit from your parents in the form of DNA to making the flesh and blood you. And so most of you have probably heard about DNA, and some of you may even know that it is a double helix and it contains all the information there is about you. And most of you perhaps have heard about protein. We're all made of protein, and all the protein in our bodies is made by our cells based on the information in our DNA. And so this brings me to the only dogma worth believing, which is the central dogma of biology, and it goes like this. Our DNA goes to RNA, goes to protein. And as dogmas go, it's actually wrong most of the time. And so, to explain RNA, some of you perhaps have not heard about this. RNA is ribonucleic acid, and so it's very similar to DNA, which is deoxyribonucleic acid. And so it's used by the cells as a messenger molecule. If you want to make a protein, you'd find the gene to make that protein. Genes are kind of like reading frames for our DNA. They're chapters in the book of your genetics. And so you find the gene for that protein, and you'd copy letter by letter, copy the DNA onto RNA. And that strand of RNA, the single strand, will then travel away to the place the proteins are made, and is again translated letter by letter into an amino acid chain that is curls up, kind of like a spaghetti nest, and this is how all your proteins are made. But this analogy really underestimates RNA as a simple messenger molecule, and it's so much more than that. And especially, a lot more happens to it between going from DNA to protein. And to talk about that, I have to bring up the Human Genome Project. You guys heard about it? It was completed in 2001, and it was to sequence all of our DNA. And before it was done, people were trying to guess how many genes we had, because they thought, one gene equals one protein, how many do we need? About 100,000? And if we had anything less than 100,000 genes, we probably wouldn't have enough protein to make our complicated bodies. And so when they finished the Human Genome Project, what they found that we actually have about 25,000 genes, which is a lot less than expected. For comparison, mice have the same number as we do. Tiny microscopic sea-free worms have almost the same number of genes as we do. Quite a lot of plants have a lot more genes than we do, and we're more complicated than onions. And so it was kind of a kick in a scientist's ego. It was like someone telling you, yeah, I know your daddy told you you were special and king of all creation, but actually you're kind of like everyone else. And in an effort to reinstate our superiority, we started looking at the other biomolecules. And what we found was that when you look at proteins, we actually have over 250,000 proteins. And you may think, well, that's great, that's way more than everyone else does. But how can that be? I just said, DNA goes to RNA goes to protein. One gene makes one protein. We have 25,000 genes. How can we make more than 250,000 proteins? There's some kind of magic going on. This cannot make any sense. And unfortunately, it's not magic, it's RNA. So you have all heard that our DNA is mainly junk, right? It's true that almost 99% of our DNA doesn't actually code for protein. And even if you start looking at genes themselves, what you find is, let's say this is a meter of gene. This is 1 meter. Only 10 centimeters, so 10% of it would be used on average to make a protein. The rest of it would have to be copied from the DNA. So you'd copy the entire thing onto RNA and then chuck most of it away. And not only is it only 10%, that 10% is all over your gene. So all these little red bits you see, that's the bits you want from your protein. So to make the protein, you'd have to copy this entire thing onto RNA, then cut out the red bits, glue them together, and then send it to be made into protein. And that's a lot of waste. That's about 90% of it just thrown away. And you may think, well, it doesn't seem to make sense. Why don't we just have that 10%, all that red bit, in one sequence in our DNA? We could copy it, and it could be made into protein. It would be a lot less hassle. And that's the way, that is the way bacteria do it. But we have an advantage over bacteria when we do it a complicated way. And that is, that is we can make more than one protein from one gene. So when the machine that cuts out and glues together the little bits that you want out of RNA, it can leave out certain bits or add in other bits, and suddenly you get a completely different protein. They're very similar protein, but with slightly different activity. And that means that you can make one protein, perhaps with this little red bit left out in your brain, and you can make a different version of the same protein with this blue bit added in your liver. And that means that our cells, because they can make these different versions of the same protein, they can have so much more control over the actions and so much more variability and just a higher function. And so, you may think, okay, but this kind of sounds complicated. Why would we ever want to find out about this? Can we not just study something else? But there's many reasons to study how this exactly works. For example, the machine that does this entire cutting and gluing together, when viruses infect our cells and they try to make viral proteins, they don't have a way of doing it without our machinery, and so they have to use ours. And it would be nice to know how we could stop that. Cancer cells, for example, make slightly different proteins to the cells around them, and we're not exactly sure why or how that is controlled. But I think most importantly, mutations, when they occur, you think for a mutation to occur, it would be more likely to occur in the 90% that is not actually put into the gene. But that 90% can affect how these bits are cut and glued together. So you have a mutation here, it may mean that all of these red bits are left out and you have a protein with no function. Or you may have a mutation here and you'd include this huge bit that would make a huge protein that would be too cumbersome to work anything, and this is how you get disease. And so it would be really nice to see how this cutting and gluing is controlled so we could have a chance of understanding this disease better. And so you think, okay, fine, it's worth studying. How would you go about it? So how would you go about studying any kind of cellular biology? And what the biologists do is they ask a chemist to do it for them. So as a chemist, I would go and send in a probe. And probes are kind of like our little spies. So other planets and cells have a lot in common. And they're very interesting to study, but we can't really go and have a look around. Planets are far too far away, and the cells are far too small. And so what we do is we send a probe, just like the Mars rover was sent to see if there's water on Mars. We designed it so it can pick up water, and we designed it so when it finds some, it can send a signal back at us. Same way we design a probe to send into the cell to look for the information we want, and then give it a way of signaling back. And one of the ways we can do that is to make it fluorescent. And so, if I wanted to go and study the spliceosome, I would find a molecule that interacts with this machinery, spliceosome machinery that cuts and glues together the bits of RNA. I'd find a molecule that interacts with it. I would give it a thing to do, so maybe information that I want to find out, and I would give it a way of giving this information back to me, so for example, fluorescence. And by making these probes and sending them into cells, we'll find out more about these molecular machines and how they work, and this gives us a better chance of understanding diseases better. Thank you.